beginning of my work on polyploidy and adaptation to salt stress, the physiological, biochemical, and genomic determinants of tolerance in citrus polyploid rootstocks. So as you might know, citrus is one of the most important food crops in the world, and unfortunately, citrus are classified as a salt-sensitive crop. But it exists a very strong diversity in this trait in the citrus genus. For example, the Pancerus trifoliata genotype is considered to be highly susceptible to salt stress, while the Cleopatra mandarin genotype is thought to be more salt tolerant. In citrus cultivation, the use of rootstocks allows a better adaptation to biotic and abiotic constraints. Indeed, the use of the Pancerus trifoliata, for example, is widely used in Corsica because it's a rootstock that can improve the tolerance of the cultivated citrus species to the Tristeza virus and the Phytophthora. However, the Cleopatra mandarin rootstock is not widely used here because the Cleopatra mandarin rootstock is sensible to the Phytophthora. But the Cleopatra mandarin rootstock could be more tolerant, could bring tolerance to the salt stress. So we decided to investigate more in the Cleopatra mandarin rootstocks. And previous work led by my university, the University of Corsica, and the CIRAD and the INRAE, has shown that tetraploid rootstocks can improve tolerance to different constraints, that is to say biotic or abiotic constraints. But the mechanisms involved in such tolerance and the exact influence of polyploidy on tolerance in citrus is not yet known. So we decided to investigate in detail to what extent can tetraploid rootstocks improve the adaptation to salt stress of freestanding plants compared to the use of diploid rootstocks. And to do this, we selected 6 different genotypes for experiment. That is to say, 2 diploid parents, a diploid Cleopatra mandarin and a diploid Pancerus trifoliata. These 2 diploid parents were crossed to give us 2 double diploid parents, a tetraploid Cleopatra mandarin and a tetraploid Pancerus trifoliata. So these 2 double diploid parents were then crossed to give us 2 diploid hybrids, and you know the offsprings resulting from the cross of the Cleopatra mandarin and the Pancerus trifoliata is called a citrondarin. So here we have a diploid citrondarin and a tetraploid citrondarin. Plants selected for experimentation were divided into 2 blocks. Stress plants were irrigated twice a week with nutritive solutions supplemented with salt, while the control plants were irrigated twice a week with nutritive solution only. Salt concentration were regularly increased, starting from 30 millimolar to reach 90 millimolar and then stabilized at 90 millimolar for 2 weeks. And to understand and identify the mechanisms involved, we developed an integrative approach, including the study of physiological, biochemical, and genomic determinant of salt stress response in citrus. So as you can see here, our preliminary result seems to confirm the previous hypothesis, and you can see that the Pancerus trifoliata rootstocks is the most sensitive genotype we tested for salt stress, as the tetraploid Pancerus trifoliata and the diploid Pancerus trifoliata are very affected by salt stress. the Cleopatra mandarin rootstocks is much more tolerant to salt stress, but we can see clearly that the diploid rootstock is much more affected to salt stress than the tetraploid rootstocks. And strikingly, we can see that the hybrids, both diploid and tetraploid cetrandarins, show almost zero symptoms of salt stress. So this is really encouraging for us because this will mean that the hybrids has potentially inherited both adaptation traits from their parents, from the Polyploidy trifoliata and the Cleopatra mandarin rootstocks. So our physiological and biochemical study is completed by a genome-wide diversity study that will allow us to compare sequence variation between Polyploidy trifoliata and Cleopatra mandarin with our transcriptomic result obtained by RNA-seq. And this will help us to identify possible cis determinants, that is to say possible variation in promoter sequences of overexpressed or downregulated gene involved in salt stress response in citrus. This work will lead us to provide new evidences to explain the advantages of tetraploidy over diploidy, and we will be able to determine if there is a constitutive pre-adaptation linked to tetraploidy, or if tetraploidy improved the response to stress through the phenotypic plasticity. So thank you so much for having listened to me. And as we say here in the Corsican language, we are looking for collaboration. So if you are interested by our work, don't hesitate to contact me at this mail address, bonin_m@universityofcorsica.fr. And thank you again for this conference.
Thank you, Bonnin. I'm sure you all enjoyed your presentation. It was a nice work on tetraploidy versus diploidy on the rootstocks. I hope you find more collaborators and you're able to add more to your research knowledge. And if there are any questions by the participants, if somebody wants to ask something, I'm leaving the forum open for discussion.
Thank you. I will be pleased to answer any question.
Yeah.